So it's important for these organisms to have these structure, the extra cell wall. It really is just for protection. Right? Now, let's look at a sample question here. It's a little bit much, but we'll go through it together. It says, the effect of water-soluble pollutants on membrane permeability. So it gives you these treatments, and here's what is going on. A student came up with this hypothesis that water-soluble pollutants damage living organisms by increasing the permeability of the cellular membrane. In order to test the hypothesis, the student investigates the effect of isopropanol and acetone on beetroot cells. The vacuoles of these beetroots contain large amounts of beta-cyanine. Beta-cyanine is a water-soluble pigment that is released into the extracellular environment as a result of increased membrane permeability. The student prepares identical samples of beetroot tissue and incubates each of these samples 15 minutes in specific solution. What does it mean to incubate? Anyone know what incubation is? Yeah, so you're just putting it in an environment with that solution, right? Okay. If you've ever raised chicks or anything like that, right? The eggs have to be incubated and keep. In this case, the incubation it's the treatment, so it's sitting in that solution, closed environment from other things. At the end of the incubation, students measure absorbance of 460 nanometers of light. What does that mean? Light comes in wavelengths, right? So the visible spectrum, the colors we see are which ones? Roy G. Good? Red, orange, yellow, right? So between red and violet, which one is the highest energy? Violet, right? Blue light is a lot hotter than, so is red light. Even though I think of red as being hotter, right? That's not the case. Why? Because blue light has shorter wavelength. Shorter wavelength means higher frequency. What in the world am I talking about? If I'm exposed to X-rays on a daily basis, I'm doing a lot of damage to myself. My chances of having tumors and cancer increase, correct? Why? Because X-rays have a very, very short wavelength. Right, guys? Okay. We're not going to use computers. So, blue light is shorter wavelength. So if I'm going to do a comparison of blue light, maybe it looks like this compared to my red light that looks like this. And then if I'm looking at X-rays, it looks like that, right? So the more number of waves pass by a given point per second, the greater the frequency. The greater the frequency, the greater the energy. We don't want to be exposed to high energy waves all the time because they damage DNA. When these go into your cells, they go and mess with the thymine bonding, so that it allows for thymine to bond with another thymine and messes up. So that's a mutation, right? We don't want that. We want thymine to bond with what? Adenine, right? So if it's bonding with each other, then it's obviously mutated. So things like UV rays and X-rays do a lot of mutation. Blue versus red, the only visible spectrum you see is this. Those are the only colors you see. Okay? Red is the longest, so everything after this are things like infrared, microwaves, radio waves, TV waves. Those are not a problem because they travel such long distances, they don't have a lot of energy. The shorter the wavelength, shorter distance, higher energy, right? So blue light has higher energy, and the range of this is about 400 nanometers or so on this end, and about 700, I think, 20, somewhere around there on this. Nanometers is 10 to the -9 meters. How long is a meter. You've seen those meter sticks, right? It's a little more than a yard. A yard is like 90 centimeters or something, right? So think about now a meter. Now take that meter and divide it 1 billion times, and you get a nanometer. Okay, so you're still talking about pretty short distances, but it's a comparative red to blue. Anyway, so on that note, absorbance of 460 nanometers of light, a greater concentration of beta cyanide in the solution surrounding the bead results in greater absorbance. Okay, so if something is blue, does it absorb blue light? No, right? So if you give plants green light, what happens to plants? They don't live. Right? Because the green light reflects, and if they can't absorb the light, they can't have the energy they need in order to go through photosynthesis. Okay, so 460 nanometers light is around indigo violet region or so. So, and beads are what? Red? So it makes sense for reds to absorb the blue. Okay, student analyzed the data from the investigation, concluded that the estimate of the mean of one treatment group was unreliable. Which of the following identifies a treatment group most likely to have provided an unreliable estimate of the mean, and correctly explain why that is? Now, remember the standard errors and all that good stuff? Okay, so it gives you their standard error of the mean. It gives you the mean absorbance, and then it says that one of these treatment groups didn't really follow what she wanted the experiment to do. Okay, so on that note, which one do you think it is and why? So your choices are there, right? Let's see if I can change this up. If I can change this. All right, so it says treatment group 2, 3, 4, or 5. Those are your options, right? So I'm going to do this thing. All right. So 2, 3, 4, or 5. First of all, which one do you think? Why? Do you see some kind of a trend of what should happen with a certain amount of water versus how much it should absorb, and what kind of standard error is seen? So like 70% isopropanol, 30% water, I have a mean absorbance of 0.164. 10% water, I have an absorbance of 0.125. And then if I have 50% acetone, 50% water, we have 0.215. 70% acetone, 30% water is 0.274. So do I expect an absorbance to be greater with more water, less with more water? What is going on here? Yeah, what do you think? So you think 2 doesn't fit? Okay. All right. What other thoughts? Yeah. Okay. 0.1, right? Compared to 0.03, 0.03. So standard error is high. There's less water, but the mean absorbance is much lower. Okay. What other thoughts are there here? Are there other thoughts besides treatment number 2 as being one of them? So is that our only option? Sure. Yep. Yeah, because the error there is way too high, right, compared to the rest of them. Okay. And we will spend some time talking about chi-square analysis and standard error and deviation a little later on. Okay. So those are the key takeaways.